to the Digital Agriculture Midday Seminar Series. Today we have, actually all the topics are overlapping. When we talk about what our topic should be, we're all proposing the same kinds of things, from different perspectives, whether it's from a row crop agriculture maybe side, or a controlled environment side, or digital forestry side, or digital agriculture side. A lot of overlapping. A lot of topics are of interest to the different groups. One of the topics that I wanted to try to highlight today was I really wanted to try to take a minute to highlight the research capacity that's been built up inside of the plant sciences initiative at Purdue over the last 10 years or so. And I just wanted to start by saying we have a really strong team of researchers that allows us to do the things that we do. And so we're lucky today that we're having a presentation from a team. I'm sorry, Jan, I didn't get a chance to go back and look at your CV, but about 6 or 8 years ago, Dr. Yang came to us from Dow AgriScience, where he was involved in, I'll be generic and just say digital agriculture, phenotyping writ large. We were lucky enough to elect Dr. Yang to join us here at Purdue. And he was really attracted. I was on the search committee, or one of the co-chairs of the search committee. He was really interesting as a candidate because he has a skill set at the time that was really unusual, which is he knows a lot about engineering and he knows a lot about plant science. And at the time, that was a bit like a unicorn in terms of the kinds of people the number of people that were out there that had those skill sets. And so we were lucky enough to get Dr. Yang to come to Purdue. And really the challenge that we put in front of him is, hey, we have this vision for phenomics, controlled environment phenomics and controlled environments of plant controlled environments, and phenomics of plants and field environments. And he agreed to come join us, and I'm going to tell just a little joke about our early relationship. Not bad, promise it's not bad. So we had just completed the controlled environment facility, the basic skeleton of what it's not nearly as sophisticated as it was today, or as it is today. And we had the instruments, we had plans, we had stuff going on. And I think, was it January? I think he started January, February, early in the year. And we still didn't really know what we were doing. And when we were looking at our candidates, we had different kinds of people. I said, we definitely need to have someone that understands the engineering of this thing, because we don't know what we're doing. We don't even know how these things talk to each other. All these devices talk to each other. How do we do the automation and actually make it work? And so Dr. Yang came in, looked like everything was going really well. One day, midday, a few months after I started, he came to my office door, closed the door, sat down. He says, I don't know if we're going to get this thing to work. Imagine that 6 years or so ago, and what you're going to see today. Dr. Yang is going to introduce his team, Sun and Libo, two of our new junior scientists on the team that are going to describe different aspects about phenotyping in controlled environments and field-based environments. So thank you very much for joining. Dr. Yang, thank you for sticking around, even though at the time it seemed like mission impossible.
Thank you very much, Peter, for the introduction. It was scary, actually, and also very exciting to have such an opportunity to work at Purdue with the investment we've made in the facilities and the plant science overall. So I'll take just a few slides to give you a very quick overview in terms of where we came from, thanks, and what we are trying to build up from the initial efforts. And I'll leave the most of the time to Sun and Libo just so they can share the real meat. Actually, they're the one actually putting it out. It's a team effort. So I'll skip this slide. So Plant Science Institute is the outcome of the Purdue Move initiative, started about 2013. Since then, the school and the college has invested over $40 million into the expansion of our capability in the basic biology research. To accomplish that goal, of course, the vision is we need to establish the capabilities in the field-based phenotyping as well as in the controlled environment, just so we can accommodate and enable the PIs, scientists, either on campus or outside to collaborate in this domain. And of course, with those facilities, we need to have very robust capability to integrate data together. And also, hopefully, we can also leverage these capabilities to facilitate and enable commercialization of the learnings and what have you. So with that vision, ICSC was brought into service in the year 2016. And this, of course, in line with the very strong support from the Indiana Corn and Soy Chertoff program. This is a facility. I'm not sure if everyone here or online has an opportunity to visit us. If you have not, you should. This is an amazing facility with a very large footage and with an array of machineries and instruments that covers literally every step of your need in terms of sample processing and measurements. And also here, we have a very strong capability that we have been providing in the last 4 seasons, leveraging this very unique Griffin UAV system for remote sensing in the field. Right now, this system enabled our scientists to have multimode remote sensing in using 5 different sensors. If you have any needs for color imaging or any needs for hyperspectral, or most recently, we have a thermal imaging system and pipeline established with the strong support from Griffin. On control environment side, we have the very strong support from Ag Anomaly C. That's why the facility is called Ag Anomaly C High Throughput Phenotyping Facility. This is a facility, to my best knowledge, the largest one in North America that's growth chamber based for phenotyping purpose. We are in any time of the day, we're able to accommodate over 600 plants in growing measurements. The measurement mostly can be done, of course, ground truthing can be done by many, but the system, what Sun and Libo will share with you is that we have established the imaging-based phenotyping system to phenotype and measure the root and the shoot of the same plant with very high resolution, both spatially and temporally. Before I leave the time over to Sun and Libo, the last thing I want to stress to my audience here is that it is really relatively easier to appreciate the effort in terms of building a facility, building a sensor, a new toy. If you buy a new sensor, a fancy sensor. That's not the point. Of course, we need more advanced technology to enable our scientists in research. But on top of that, a more important, if not as important, if not more important aspect is the capability to extract information out from the sensors, the imagers that can accommodate, enable our PIs, our students, our postdocs in their research. For that matter, our team has never really stopped this effort, continuously trying to improve our algorithms, improve our methodologies in doing a better job to extract the information out of our sensors. And so today, Sun and Libo will have a chance to tell you their experience, their learnings on either shoot imaging that's mostly come from Sun. Sun is our infrastructure data scientist for the last almost 3 years. Libo joined us as a postdoc about 2 years ago. Libo focused more on the root end, and Sun takes care of the whole facilities operation in terms of data processing. But today his story mostly focused on the hyperspectral imaging. So without further ado, Sun, if you could please.
Hi, this is Sun. So here's a little more slides about why you want to work with a plant science institute. So first is you can do the exact same experiment over and over with slightly a little bit of differences in your treatment, in your plants, and so on. And we also have next generation sensor that will detect any minor detail, what's going on in the plants, with your plants. And whenever you have concerns, doubts, or anything you want to add on to your experiment, control, soil amendment, whatever, you can come to the facility to do your own thing. And then whenever you have issues with data handling, data management, data processing, you can come to us to work with us. We're experts. So here's the detail of sensors, but the takeaway from this slide is that first, we have a high throughput phenotyping system. And the second is you can get a timely measurement of your plants in a regular basis. This is sometimes sometimes it is not achievable in the outdoor environment. And then, although, yeah, just like the outdoor environment, we deal with the plant growth, data collection. Okay, the outdoor built environment, yeah, they sometimes don't deal with the data acquisition, but we also get all the we collect all the data and do the data processing. And what you can do is you can just focus on your research. For example, designing your experiment and doing an analysis and writing your paper. So here is the basic workflow, how the hyperspectral data is processed to produce a plant phenotypes. The hyperspectral data, in a nutshell, is basically a layer of images. So each layer has a reflectance values in each pixel. And then each layer represents a layer of image or layer of reflectance values in different colors, which can be blue, red, Purdue yellow, St. Louis red, whatever, and stuff like that. That all the way up to from blue color to the infrared. And then we select one of these layers to for further analysis and separate background and plant pixels. And then we separate region of interest. For example, we can think of the whole plant, upper structures, lower structure, and so on, so that we can focus on specific part of the plant to get the plant phenotypes. And then we get spectral information, which typically has two types of information. First is reflectance values, and the second is vegetation indices. So, as I said, there are two types of plant phenotypes that is derived from the hyperspectral data. The first is reflectance. So I share about what is the information in the hyperspectral data. So each layer will have the reflectance values in each layer. And if we take that out and get an average of it, we'll get an average reflectance value in each layer, or aka each color or wavelength. So for example, if we get an average reflectance from this layer, we would get average reflectance in 400 nanometers. And from the next layer, we'll get an average reflectance in 401 nanometer, and so on. So that if we stack them all together, we get a reflectance curve like this for each plant or each measurement. And the second type of information is vegetation indices, which can be sometimes a proxy of plant vigor, plant resistance to the disease, or weakness to the disease, whatever. It is not very it is sometimes very ambiguous. It needs a lot of data analysis to compare with the ground truth data, but here's what hyperspectral data analysts do. So, for example, I'll make an example with the most widely used vegetation indices called normalized difference vegetation index, which is sometimes similar to plant vigor. So it uses a layer image layer in 607 nanometer and 800 nanometer. So we get two of the layers, and then we do image band analysis, image band map, and the result will be still an image. And then we do the same thing with the region of interest to get, for example, average or standard deviation or percentile, and then we get a number. And we do this for 50 different vegetation indices definitions. So what you get is this kind of very wide table. And optionally, you might also take got some browser data in the facility. So you can do whatever you want to relate these data and this data. So here are kind of a generic workflow you can do after you get all those input data sets. You can, based on your research needs, you can do predictive analysis. You can do kind of find the cause of what happens, and because what happens, what makes the plant response to the different control treatment. You can just want to just see the overall trend in a graph or something like that. And then you can choose method, basic a broad method, like you might want to use the conventional method used in your specific domain, or you might just want to use descriptive analysis. Maybe you want to use the ANOVA or machine learning, deep learning, whatever. And then maybe your graduate students or postdoc will do the real things. But whenever you have inquiries or concerns about doing the in-depth analysis, you can come to us to say, hey, can you help us with the data analysis with your data? We can help with that. So here are three different use cases using hyperspectral data. The first case is with about stress with water. So when we look at the corn leaves in the later stage, the lower leaves senesce earlier than the upper leaves. So we can ask this question, okay, this kind of lower leaf senescence will be more pronounced when the water stress in the water stress plants. So yeah, we did analysis using this kind of experiment settings, and here's the result. We just used a simple exploratory data analysis, which generates box plots with different dates using plant vigor index, which is NDVI I explained earlier. So if you look at the plant vigor index versus data after planting, you can see the upper leaves, plant vigor is almost same and decreased a little bit at the end of the season, but the lower part, in the lower part, the plant vigor increased and decreased much faster than the upper structures. And this kind of trend is more pronounced for the dry down plants. So here is the simple result you can just get just by plotting the data in box plot. This is raw data from the entire sheet, one of the column. So here's a very simple example you can do with the raw data. And the second data is a little bit uses more advanced method, I would say. So this was led by Lori Hoagland's lab. So cadmium is one of the heavy metal element that can be super accumulated in plants, and it can be toxic to the people, sometimes causing itai disease, means it hurts disease. So their team came to us and asking, does the vegetation indices data or hyperspectral data really, would that be possible to really find the difference in the cadmium content in the plant? So we did the experiment with 3 different cadmium concentration in the soil, and as well as control. And it was the result. We don't have the beautiful graph for this case, but we did because we did the ANOVA. And then they were able to figure out which indices were useful to differentiate different contents of cadmium in soil. So these were the list of indices, which was grayed out because you don't need to read all that. But if you look at the raw images, you can see the control plants. They are very strong, healthy plant fiber in the center, and overall, the red means more strong in this case. But for the case with the 15 ppm cadmium in soil, there are more bluish pixels here, especially in the vein in the kale plants. So here were minor details we could find in the hyperspectral data. And the third case was using even more advanced method called the machine learning. So this case, we did an investigation about soybean sun death syndrome, which caused mostly damages the root. And then when the symptom is seen from the foliar, the leaves, there's nothing we can do. We can just abandon the whole field or whole area around that. So they're very devastated, very a disastrous disease. So here's the result. We did the machine learning classification for this and only used the principal component analysis just for visualization. And when we look at this, the data, the disease plant, healthy plant are kind of separated well. And the result was this. Even in the absence of foliar symptoms, we could 75% differentiate those plants. So here's the result. And the result was published in Plant Phytofrontiers this year, just a few months ago. And here's the slides for the X-ray CT analysis stages here.
Hello, everyone. I'm Libo. I'm the postdoc in the Institute for Plant Sciences team. Today, I'm going to talk about a case study, impact of computed tomography (CT) X-ray dose on corn root and root development. So by this case study, I'd like to demonstrate how we can use the X-ray CT imaging system to non-destructively observe the growth of corn roots. First of all, is the background. So the root system architecture plays an important role in the uptake of water and nutrients. So studying and analyzing the different root trees could be a promising way for us to improve the corn yield. But the traditional methods to measure the roots need to dig the roots out of the soil, which usually is destructive, time-consuming, and labor-intense. Instead, the X-ray CT imaging technology enables non-destructive and three-dimensional observation of corn roots. However, a significant concern was that the X-ray radiation could do damage to the corn plants. For example, the team did an experiment in 2018. So they found under the X-ray radiation less than 8 gray, the total root length of fibra bean was reduced significantly compared with those in control group. But this phenomenon was not observed for barley plants. They were under the same experiment setting. So the susceptibility to X-ray dose is dependent on the species and variety. Also, the previous literatures or papers focus on the impact of X-ray dose on roots or the environment near the root. For example, the microbe near the root. Field reports discussed the impacts of X-ray dose on both of root and shoot with a combination of X-ray CT imaging and hyperspectral imaging technologies. So that's why we wanted to fill this research gap and conduct this study configuration of the imaging system. We grew the column plants in one of the growth chambers at APF with a precisely controlled environment. And then the imaging of the plants went out of the growth chamber and went through the hyperspectral imaging system to capture the hyperspectral images from the top view and side view. After that, the plants stop in front of the X-ray CT imaging booth. And inside the imaging booth, with the X-ray tube on the left, the plant sample in the middle, and the CT scanner on the right. So the X-ray radiation penetrates the soil and the roots, and the CT scanner capture the signal to generate a 2D projection. Also, the plant sample rotates in between of the X-ray tube and the CT scanner, so that thousands of these 2D projections were collected for one sample. Later on, we can reconstruct the 2D projections to a 3D structure. About the experiment design, in total, there were 28 column plants with B73 times MO17 genotype. So the plants were divided into 4 groups: W1, WS1, WS3, WS7. WW means well water, WS means water stress. Because of the water treatment difference, there is a slight stage difference before harvesting. So for W1 and WS1, they were scanned only one time before harvesting. For WS3 plants, they were scanned 3 times at V5, V8, and V9 stages. For WS7 group, the plants were scanned 7 times from V3 to V9. On the last sampling day, all the plants then moved out of the growth chamber and went through the hyperspectral imaging tower to capture hyperspectral images. And after that, I harvested all the tissues above the soil to measure the shoot dry biomass. So after that, all the pods were scanned in the X-ray CT imaging. So at last, all the pods were disposed properly. So for the W1 and the WS1 group, they were scanned only one time on the last sampling day. So it means the X-ray radiation did not have a chance to take effects on the plants. So the effective X-ray dose for them is 0, and they can be regarded as the control group. For WS3 group, the effective X-ray dose for them is 1.2 gray. For WS7, the effective X-ray dose is 3.5 gray. About the canopy hyperspectral imaging process, the data actually is from Sun, my colleague. As shown in the figure here, for each plant, the spectrum was extracted. And the spectrum is from 400 nanometer to 2,500 nanometer. So based on the spectrum here, we can calculate multiple vegetation indices. And 5 of them are listed in this table here, which cover the visible bands, the near infrared bands, and the short wavelength infrared bands. For example, the widely used normalized difference vegetation index, NDVI, is related to chlorophyll. So we use these multiple vegetation indices to compare the difference between the different active treated groups. Next is the root city image process. As mentioned earlier, the 2D projections were reconstructed to the 3D structure. So as shown in the middle figure here, it was segmented from the soil and the pot. So it's the 3D root system architecture. And after this, we did skeletonization to get this skeleton image here. So in this skeleton, we know how many voxels it has, and combined with the voxel resolution, we can calculate the total length. Also, other root trees, for example, the maximum root depth and the root number, the root angles, and root volume, also the root volume convex hull. And also with these root trees, we can compare the difference between different activators groups. If we want to compare two root structures, we can put them side by side, like we did here. We put them on the top. So the left one is from well water group. The right one is from water stress group. So we can see the left one, the structure is a little bit larger than the right one. On the bottom, there are two figures of skeleton images. So we can also calculate the root radius and map the root radius data on the skeleton images so that we can check the root radius changes with depth. For example, here, for the well-watered plants, the root radius becomes smaller from the top to bottom. And at the bottom, the root radius, based on the color bar here, is around 0.5 millimeter. So the root diameter is around 1 millimeter. And at the top, the root radius is, some of the roots is around 3 millimeter. So as mentioned by Dr. Yang, they may be breathing roots. So usually they are larger. So if we go to the water stressed plants, same story, the root radius becomes smaller and smaller from top to bottom. At the bottom, the root radius is around 0.4 millimeter. So the root diameter is around 0.8 millimeter. Also at the top, the root radius is around 2 millimeter. So for the, if we only compare the root thickness or the root diameter, the water stressed plant is smaller, it's less than the well-watered plant. I think this makes sense because there is a stage difference between them. Also, I manually dig some roots out of the soil and measure them. So the value, I think the value they match, but we need to get more ground truth data later. Also, we can check the root number changes with depth. So for each plant, we can get one root number changing curve with depth from top to bottom. And in each treatment group, we can average the curve into one average root number changing curve as shown in this figure here. So the root number increase slightly at the very beginning and then decrease. For the water stress groups, WS1, 3, 7, they mix together. But there is a clear difference between the well-watered treatment and the water stress treatment, especially at a higher root depth, for example, 250 millimeter. So you can see here. So typically people use total root length to compare different treatments or different hybrids. But one problem with the plants indoor, because we grow them in pots. and the total root length is limited by pot size. So the root number at a higher depth could be a better alternative to the total root length. We also did a ANOVA test on all the root and the shoot trees. For example, on the left is the total root length data set. So based on group means, we can separate well-watered and water-stressed treatments into different classes. The well-watered, the plants, they belong to one class. The water-stressed plants, they groups, they belong to another group, which means there is no significant difference between the water-stressed groups. So the different X3 dose didn't impact the total root length in different X3 treated groups significantly. So if we go to the shoot dryer biomass dataset, the story is the same. Also, we use the multiple shoot vegetation indices, for example, NDVI here. So also the same story. So conclusions here, number one, the different X-ray dose did not significantly impact root and shoot development in this study. Number two, our X-ray CT imaging system at APF enables our users or researchers to non-destructively conduct 3D imaging of corn roots at multiple growth stages. Future opportunities. So if we use we want to reconstruction of the raw 2D projections with a higher resolution, we can detect more finer roots. For example, here in this figure, we can see more lateral roots. And my next step is to build hierarchy for different types of roots. For example, primary roots, secondary roots, and lateral roots. And calculate root trees, such as total root length for these different types of roots respectively. So next time when we build correlations between root trees and other trees, for example, yield, we can only use the trees main roots or the lateral roots to build the correlation with yield. So by this way, there could be a better chance for us to observe a higher correlation. So with our X-ray CT imaging system, we can do root phenotyping not only for corn, but also for other species, such as soybean, roots, pear, also tomato, and sorghum. I think that's all from us today, and here is our team. Yeah, thank you very much.
Yeah.
So, is there a minimum plant size or a stage of development for X-ray? Like how small plants can be to be able to detect the roots?
Okay, so the minimum, the maximum resolution we're using is around 100 nanometer. So it's 0.1 millimeter. So if your roots or your target is is larger than that, we could detect that. So
Depending on your crop.
Yeah.
If it's corn, we can push to V3. So if it's corn plant, we can push to V3. That's the earliest we can be comfortable in terms of providing you that. If it were soybean and tomato, normally we do it at week 3, week 4. We should be very comfortable in terms of providing you data that could make sense to you.
I have kind of a follow-up question to that. So
Yeah.
I understand that like the amount of moisture in the soil and the moisture in the roots, can impact both resolution of the X-ray sensor. So what I was wondering is like, where does that level of soil moisture fit in with the different levels of like well water, a water stress 1 through 3?
Yes. So, yeah, for well water. So for well water, actually we based on the a saturated soil weight to decide well water and water stress. For well water, we use 50% of the fully saturated soil weight. For the water stress, we use 20% of this weight.
And so at 50%, you're able to get good, high resolution images that distinguish between the soil and the roots.
Oh, no. I was talking about the well water and the water stress, how we determine them.
Yeah.
But when we do imaging, we need to dry them down below 20% to get good quality of root images.
Okay, but it's only for a short time period and then high they can get versus the things in this experiment spent the whole time that there's different water levels.
Yeah, so that's why before the last sample day, we need to dry them down like for 2 days to do the final experiment.
And has there been work to look at like what that does to plants? Do you suspect that has any negative implications on the plant growth?
That's what is a medicine we have to swallow.
Okay.
Definitely, we cannot rule out a, negative impact if you dry it to like below 20%. 20% is already a drought stress, but it needs to be even drier for the machine to have a, high quality, the imaging capability. So for us to say there's no impact, it's very hard to make that argument. With that being said, though, this impact is transition anyways. So we are not treating the plants in a long period of time.
Okay, so it's minimal and.
Yep.
Thank you.
Yep, sure.
Well, I'm fascinated by your root imaging, and I was curious, what's your accuracy level? I was wondering, like, if crop residues from previous crops or earthworms, or perhaps differences in soil texture, a sand versus a clay. How does that affect your results?
You want to give it a shot?
So for our experiment, we just use the very commercial soil like a Sungchan Oh. Did we try sand before? Okay,
So the impact of soil texture definitely is a factor we have to handle. Depending on the type of soil, actually the machine itself comes with 3 different layers, levels of radiations, just so we can, the radiation can penetrate. For heavy soil, you have to have a higher level of energy, just so it can penetrate the pot. For like the peat that based potting media, we normally use a relatively lower energy. So the texture definitely plays a game and plays a role in our process of the image acquisition. Yes.
And could you do this in the field? I mean, how deep could you go into a soil in the field? I can imagine not very deep.
Not that we know of.
Yeah, okay.
Up to this point, we do not have knowledge in terms of a movable CT.
Yeah.
There is a movable, what do you call it, MRI machine. But in essence, it's still an MRI. They just made it small enough they can move it across a field. But you still need to dig up the whole basically volume, and then and then to shove that into a chamber,
like a tree spade or something like that. And okay.
There's a team in Texas A&M or Denver Plant Science Center. They're making some kind of like in situ MRI or ground penetrating radar system to see
Yeah,
the
very nice.
system.
Question, this is going way back to your experimental design. You talked about the low water plants were sampled at B10, I think, and all the other plants were sampled at B9.
Yeah,
Is that because they were grown the same amount of time and they happen to be different growth stages? Or what was the rationale for the difference?
water treatment. Yeah, because, why water today, I mean, well, 50% of the total, the fully saturated soil weight for the Southwest, always came to 20%. So there is water irrigation difference.
Sure.
Yeah, at the same time.
So that's my question, same time, WWS were grown for the same number of days.
Yeah, totally the same from sowing to harvesting, totally the same.
When you talk about the root depth,
Mm.
so how you guys calculate that? So it's just looking at, because you are using a pole like 60 centimeter depth, somewhere there. So how you say, okay, this is the maximum root depth?
You mean here?
You are just tracking like over time, how
Mm.
getting the root, different the profile.
How do we know the depths?
Yeah,
So actually it's based on the scanning image here. So after this, we can make this, the layer of here equals to 0. And then counts how many layers we have. Actually, each layer is one voxel, like a pixel level. So we can count how many layers. Then from 0 to how many layers, we know the depth, and also we know the voxel resolution. So yeah, then we can know the depths.
Yeah, in the earlier stage, we can see the growth of the root in the vertical axis, but once it reaches a 40 centimeter
Okay.
bottom,
Yeah.
it is really not very differentiable how long or later.
so that will happen about 20 days more or less, or less than that.
I think at V5 we can touch the bottom already.
Yeah.
So yeah.
No,
Your experiments had multiple doses of radiation given. Were they dried down for the 2 days before
Mhm.
That scan, or they were kept at their water consistency and then hit with the X-rays throughout the experiment?
On the last summer holiday, we did not like water them again. I mean, they all in drops situation to go through the.
Yep. But on the treatments where you gave them doses of X-rays multiple times, did you dry it down?
Oh, got it.
Did you do that or
We
no?
don't need to.
Okay.
So we did the dry dancing only because we want to get good quality of imaging. But if we don't need see the views very clearly, or we just want to explore the views to X-ray radiation, we don't need to dry them down.
Okay.
Yeah.
And the water doesn't attenuate the X-rays too much. It shouldn't. Okay. That's it.
I don't think so.
No. They do cause trouble for us to do analysis,
Yeah.
but whatever exposure, the roots, they are still exposed.
Okay, super. Thank you.
So first of all, thank you for your wonderful presentation. I have a question about the X-ray root scanning. So can your system now handle field soil?
We can,
You can handle.
we can, certainly we can. We are more than happy to work with you for the type of soil you have in mind. It's not like we can have a turnkey, just like right away we can give you the answer, but we are always really willing to work with you. We may take some tweaks, algorithm-wise and also operation-wise, but we believe this machine is good enough for a quite wide variety of different soils.
Yeah, I
So we
mean,
just
your
need to figure it
system, last time I think we talked, you mentioned that, the field soil might be too heavy.
Yep.
For your
Yep.
system, right?
Yep,
So,
that's for the belt. The belt, when it carries for each single part, there's a weight limit. That's
Okay.
the heavy, that's the That's real, what's say, the mass. There's also another heavy is the texture to happen. So just for the signal to have the chance to penetrate. That's another way of playing its head. But again, we are more than
Yeah, so, I mean, last time I talked to, I remember you said that, you know, because of the fuselage is heavy and the belt could not, you know, support that. Is there any technical improvement that we can do to the belt system
we
to address
definitely,
that?
we really are leaning for the so-called heavy dose, just so we can handle some more what do you say intense texture. That's one, and two, just so you know, I'm working with Fraunhofer just so we can do. No, we cannot do yet. I'm trying to make it happen just so we can eventually we will be able to scan the roots that have been dug out. So the one before we tried for you because they are not surrounded by any soil. My signals were too strong. The signals simply penetrate the roots. So the sensor couldn't pick them up. I'm also working on that, just so we can do also those type of signals.
Thanks.
I see.
But thanks for your question.
If I wanted to fly some fields at the agronomy farm,
Mhm.
how do I request science to do that?
You can come to me or go to Jason directly. Once we know your needs, we can make an arrangement.
And how big of a field can I fly in one? How big of an area?
We can fly anytime the machine lasts about 20 minutes. That's quite some significant field. But we have multiple stacks of batteries. We can do more actually if needed.
And do you, Sun, do you have processing pipelines developed for all of those sensors that you presented in your
Yes.
side of the presentation? I think you had RGB, LiDAR, Wiener, SWIR, thermal.
Yeah.
Was there anything else? 5?
There are 5. So we not only have a pre-processing pipelines for youth data, but we also have the pipeline for kind of getting plant phenotyping, and we're all actively developing how to analyze the data to relate with your ground truth data as well.
Is that pipeline public? We can use it or just we need to invest?
We're trying to make it public as much as we can. We're very actively working with Ag Data Services team out there to make it more usable for plant science scientists. But yeah, it'll take a little bit more time.
It's coming. It's coming.
Yeah, but if you work with us and if you send us the data, if you have some new data, we can process it for you.
Okay, nice. I have a few update that can invest.
If you have more interest, Sun is here and I'm here. We are more than happy to connect with you. So he is making those pipelines coming to the point that if you want to run on your own, you are more than happy. You are more than welcome to. But for the imaging end, more likely than not, we have seen our customers rely on some more than themselves. But for deeper data mining and everything, whatever pipeline you need.
Thank you.
What if I only have a little computer that can't handle the heavy duty software? What do I do then?
Okay, that's a question.
What do I guess? What do I do if I have big data but not enough computing resources in my lab? Is there a way to work with a little computer? Is there a way to work with plant science and in terms of computer hardware to process data?
Yeah, we have all the computing resources and data storing resources in our account. So one way you can do that is you can send us data and let us process send you back the process data back. The second way is you could use a cyber infrastructure, national cyber infrastructure that is named as cyber. If you pay $200, we can share our data pipeline. You can use in the cybers and you can process on their own from there. And it has a lot of data storage spaces as well as a lot of CPUs you can work with.
Internally, we have high performance computing cluster. Actually, both Sun and Libo process their data when needed on our high performance cluster. So that's a resource, it's also available if you are interested.
So all of the image processing software is also done in high performance computing, or is that some of that, local desktop?
It really depends on the size of the data and the level of detail you want to go into regarding the image processing. But yeah, we can provide any solutions if you'd like, depending on your level of expertise, level of where you want to go too much really deep, or just give a very superficial or very simple but powerful data.
And just because I'm fishing for an answer.
Yeah.
The plant science also has two systems out at the agronomy farm that can be used by any of the users to process all of their own data. You don't have to rely on your own computing. You have local compute resources, high performance computing resources, and using Cyber's infrastructure for data processing.
Thank you.
And Sun is actually really helping to build out the Cyber's component of this, so that it makes it easier to share data. Internally, it's not a big deal because there's a lot of easy ways to share internally, but a lot of times sharing externally is not so easy. And Cyber's is a really great environment.
I'm the person who says data team, especially Nathan and Betty, to can you help with us with the Cybers? But yeah, here's the time I get credit.
Julie, you have a question?
I think that I'm just so impressed by everything that's going on with you guys. It's, back to what Mitch said at the very beginning. You've come a long way, and the equipment is wonderful, the maintenance is wonderful, but what really makes something wonderful is the resources of the people. And I can really see that you make it look easy. I couldn't imagine not using it. Be able to get my plants data calculated.
Yeah, we hide all the details. Yeah, make it simple.
In case anybody's wondering, I had a plant science PhD recently collect and process all of their own data from end to end. And this was actually the challenge that I put in front of Jan when he first arrived 8 years ago. The day that we can put a plant, not an engineer, a plant scientist in front of the computer and process all their own data, we will have gotten to where we really want to get. I mean, so that the scientists have access to their own data. That's not to say that we don't love Sun, we don't love Libo, we don't love Jan, we don't love our engineers. We just want to be able to, for a lot of the routine processing, we want to be able to.
They can teach you how to do it.
Yep. Great job, guys. Nice.